Alrighty, so today we're going to talk a little bit about how you might approach a science passage. It's going to be different than approaching a cars passage for sure. With cars, you're going to have to do a lot more reading. In science, actually, what I want you to try, and I want to suggest everybody tries this at least once, is there's oftentimes no need to actually read the whole passage at any given point unless there's not any figures. If there's no figures, you're probably going to treat it more like a cars passage. But with figures in science passages, that's where a lot of the questions are going to come from. So you don't really need to put that much effort towards actually digging through the passage itself. So some of the suggestions that I have, let's start with just getting the feel for what's in the passage. Don't feel that you have to dig in and read everything and understand everything until you actually get to a point where you need to know what is being discussed for a particular question. So what I would suggest is go through and do some highlighting, do some casual highlighting, so to speak. You don't need to like highlight every single thing or, you know, identify meaning. It's really just get a sense of what's there. Look for abbreviations and like keywords. So say in this first sentence, what might pop out at us in this first sentence? Don't read it either. Just like what pops out as things I might want to highlight here. Nothing? It could be nothing. Is anybody hearing me? Maybe I've disconnected. Oh, so people are hearing. So I'm just looking for the first sentence, not the first paragraph. So maybe you guys were reading through the first paragraph. So yeah, I would say the abbreviations. Like just noting that actually would be a kind of good thing because this is going to tell me this is where I'm going to define these things. Okay, so I've got CDR there. What about in the second sentence? Anything that might jump out at me as something I might need later on? In the second sentence, not paragraph, sentence. That's okay. We're not there yet. That's what I think is what's happening. Specificity. I wouldn't, but I can see why you might. I don't know if I would highlight specificity because it's like I know it's a word in science-y passages and things like that. I would probably just highlight the abbreviation again, the FAB, because maybe I got to figure out what FAB means later on. But you know, if it's a word that you're not sure on or something like that, it might be something to highlight. But what do you see me highlighting? Am I highlighting chunks of passage or am I highlighting individual things? Highlighting individual things. I don't want us to get caught up into highlighting entire sentences because how many people have experienced this? Like you probably have one of these textbooks from freshman year. You got out your highlighters and your Post-its and all that stuff, and you started reading your textbook and you highlighted the bejeebers out of the first chapter, right? And then you've realized, like, did it do you any good to highlight that much? And so, yeah, probably not. And so you just stopped highlighting. So like maybe the first couple chapters look like that, and then the rest of the book is completely blank. And so what that is it's over highlighting. And when you highlight so much, you don't actually emphasize anything because everything is emphasized, and so therefore it's all equal. So you got to be careful with the highlighting tool on the MCAT as well, because I don't want to get into that like everything is important phase of going through my paragraphs. What we're doing here is sort of triaging, and that's why I call it triaging, the passages, is we're going through and saying, okay, if I find this somewhere else, I can quickly jump back to the few things that are highlighted and find it very quickly. That's what these are for. That's what highlighting really should serve as, is ways to find information fast. What about our third paragraph? Anything there? Or sorry, not third. Now I'm saying paragraph. Third sentence. Sorry, third sentence. CDRs are more hypervariable. Antibodies? Okay. I wouldn't, because antibodies seems to be the topic sentence. But again, if you wanted that quick reference back to where they're talking about antibodies, that's a good thing to highlight maybe. I highlighted hypervariable. That's what I went for. The hypervariable, because it's kind of a unique term and it's telling me something. So being a unique term is why I picked it. CDRs, I didn't highlight that one because I've already highlighted CDR in my paragraph. So I know it's talking about those here. Okay. What about second paragraph, first sentence? Okay. Topic is B cells. We're talking about B cell maturation. Anything else? I didn't do heavy chain, but I think it's okay. I think there's something more highlightable in this sentence, though. The V part, yeah, VH, JH, and DH. Because it looks like those things specifically are probably connected to figure 1A. So that's where, if I'm using figure 1A in a question, then I'm probably going to have to come back to this sentence to figure out what these things are. All right, what about the next sentence? We got a couple more V's that are different, VL and JL. And since we highlighted heavy chain, I might just say, okay, that's light chain. So already, even without reading this, I'm starting to see a connection. What does the H and VH probably stand for? Heavy, and the L and VL probably stands for light. So it's like, I haven't read a thing. I'm just looking at words and using the highlighting tool to pick things out. And this is why, because you can start to get a feel for what some of these things actually mean without actually reading the whole thing. What about the next sentence? To further randomize. Yeah, junction sites is kind of a unique thing. Okay. Anything else in that sentence? Maybe randomize. I think at this point I've noticed randomization is what does that word combine with from the first paragraph? Yeah, the hypervariable. So I can catch that, okay, since hypervariable, it must mean randomization, because those words mean something similar. All right, and then how about our last sentence there? 3 different mechanisms cause random nucleotide alteration, blah, blah, blah. Definitely figure 1B. And I said nucleotide alteration. Yeah, that's also what I would highlight, because that's probably what they're going to show me in figure 1B is this nucleotide alteration. Okay, great. Let's keep that process going. What about this next paragraph? First sentence, next paragraph. P addition, because that's something new. I don't know what that is. Do I need to highlight the palindromic sequence addition to go with that? I wouldn't, because it's right next to it. It's like it's right there. It's its neighbor. So I would say, okay, we got P addition, but I'm just, if I see it somewhere else, now I know where to find it. That's what I'm doing this for. That's really all I went for in that sentence. What about the next sentence? So there's some kind of repair pathway that's involved in it. Okay. And then the next sentence, it looks like Vanchika was going on to say, in addition, I like that too. What about my next sentence, the lastly sentence? Lastly, in a phenomenon. Yeah, that's a new term, I can't say that word, junctional flexibility. So I have a tie back in the next sentence, these random. Anything that ties back? Yeah, we saw nucleotide alterations before. And so this is a throwback to that previous paragraph. And then I do see that this thing occurs twice. New non-productive rearrangements occurs twice. So if I have non-productive rearrangements, that seems like something I would need. So that's what I would highlight in this particular paragraph. Do I understand what's going on? Do I know what N addition and P addition is yet? Nope, I didn't read it. I didn't pay attention. How about the next paragraph? What do you think is good in there? So they're defining a new thing, somatic hypermutation. So this paragraph must be about somatic hypermutation. My next paragraph. Or sorry, next sentence. For me, I didn't see anything that piqued my interest in that next sentence, because what are they doing in the sentence? They're just giving me a more in-depth definition of somatic hypermutation. And since somatic hypermutation shows up back to back, I didn't actually write or highlight anything in that one. In a process sentence, yeah, I went with affinity maturation because it's another process they're defining for us, and they're doing that through. And then our final sentence, those that have a weaker affinity. Apoptosis, so it's telling me that weaker affinity falls apart. The cell will die. Okay. Still don't know what the heck's going on, but I'm just finding terms. How about the figure? What do you do with figures? What should you always read on the figure first? Always, 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 always. Can't say this enough. Read the captions, whether it shows up before the figure or after the figure. Read the captions. It's one of the sort of cardinal mistakes I see so many people make when they do this, do triaging passages, is they forget that there's really important information in there. Sometimes it straight up tells you answers to questions. Sometimes they put information like, quality of the data, like they'll put a p-value or something into this figure. So make sure to read that. And I highlight it too. You can highlight on the captions. You can't highlight the figures, but you can highlight the captions. And what I'm going to do is I'm saying, okay, here's things that look familiar that I saw already. And there's fab of the heavy chain, randomly signature, create CDR3. So then they're showing me P addition and N addition, followed by junctional flexibility, further increase the hypervariability. So you see how this ties in very well to that first page. Like almost everything we saw, or actually in the whole passage, everything we saw in this whole passage ties back in. The second paragraph and the third paragraph are heavily linked to that figure. And now we know that because of the figure caption. And then glance at the figure. What is it telling you there? What does part A tell us? How many VHs are there? More than that. More than that. 44. Because you got VH1 and then a ... And I know it's a little hard to see the ... on the screen, but ... all the way to VH44. How about how many Ds are there? 27. How many J's are there? Okay. And this is what I do when I'm looking at a figure. I'm just trying to figure out what kind of information can I get from the figure. I'm not trying to unnecessarily understand what the figure means. I'm just saying what kind of information can I get from it. So I would see that I can get numbers of these different fragments. Then the next part shows me what P addition is and N addition and junctional flexibility is. So those three things are represented in the figure as well. It gives me an example of each of those different types of processes that we saw in the passage. So, okay, there's a good amount of info in that. Then that's what figures you should do at first. So all in all, how much time should this take you to just triage the passage? Not read it, but look through and find these key things. They're going to help you answer questions. That's always the big question. How much time should it take? What do you guys think it would take you to do that? A couple minutes. If you truly do it without reading, you could do this in about 1 minute. But it's hard to do it without truly it's hard to truly do it without reading. And that's something you got to train out of yourselves a little bit. Is there's reading and then there's skimming. And skimming is a very different thing. Skimming is me just looking at individual words and trying not to pay attention to everything that comes in between. And so if you can master this process down to a minute, what it does is it gives you so much time to actually answer the questions, because now your main focus is not on the passage, it's going to the questions and saying, oh, I can use the passage for this, or I know where in the passage I need to go read for, or et cetera, et cetera. Because like if I look for my questions now, first off, what kind of a question is this? Is this a 1, 3, or 1, 2, 3, or 4 of the SIRS? And let me remind you what that means. 1 is just basic retrieval or memory. 2 is the harder questions. 3 is the experimental design questions, and 4 is the data analysis, the looking at the figures questions. What kind of a question is this? This is totally, and I wouldn't know it's a 4 except for the fact that I just looked at that figure and I know, oh, hey, there's some recognizable things here, right? There's 44 V's, 27 D's, and 6 H's. Oh, that's in the figure. In fact, I could probably call this a 1 because I probably don't even need the figure for this thing. But I'm going to say it's a 4 because it's really technically analyzing the figure. So if there are how many potential sequences can the antibody heavy chain have? So this is a bit of a trick question, but let's Sesame Street this one. Which answer is the Sesame Street answer? Yeah, D's the weird one. It's the odd man out, so to speak, right? And so, should I keep it or should I get rid of it? Is it really, really right or is it really, really wrong? It's really, really wrong because what was in the passage that tells me this was really wrong? What's the whole passage sort of been about? What are some of the words we caught in that passage? Hypervariability or randomization. It's not going to be 1. It's not going to be 1. That's just not going to happen. So let's get rid of that. Let's get rid of our Sesame Street answer. And then we've got what I usually refer to as my high, low, middle. So which answer is it probably not going to be? Is it going to be the high one, the middle one, or the low one? That's probably the wrong answer. Almost always the middle answer is a trap. It's a trap. I'm very suspect of answer choice B, not to the point where I'm willing to completely count it out, but I'm pretty sure it's not going to be B, even if I thought it would be B. And some people, when you look at this question, does it look like you want to pick B? Anybody here like going, but it has to be B? Yeah, it's a very attractive answer for a question like this. But what ends up happening? What did we see? Is this VH, DH, JH thing the only thing we saw in this figure that creates variability? No. Once you do that, then these other processes, P addition, N addition, and junctional flexibility start to occur. And so what's that going to make my answer? My best answer is A. It's going to be more combinations than that minimum number of combinations. So what you see here is, and that's why X equals is so tempting, because you're like, if you're not thinking about the rest of it, you might actually go with the 44 times 27 times 6 because that's just general probability kind of stuff. But in this case, there's other things happening. And again, how did we know this? We know this because I looked at the figure and the figure showed me there's these 2 different processes going on that create hypervariability. So the lesson learned on a question like this is I didn't have to read the passage. All I had to do was look at the figure, and I don't even necessarily understand that figure yet. And I can still pull and extract useful information to answer questions from it. All right, how about this kind of a question? Which B cell process would one expect to occur in a germinal center of a lymph node? What kind of question is this? One, two, three, or 4? This is a 2. I'm not going to do it yet. I don't like doing 2 questions yet. So the number 2 type questions, we're going to skip that for now. How about 3? One, type 1. All right, I'll go for that. Let's do it now. I think it's more of a 2 myself, but I'll go with 1. It's more of a like go back to the passage. So what should I do at this point? How am I going to approach this? Can I answer it right now from my own knowledge? Might take some scratch paper, but I also got to figure out what P addition is doing, right? So what should we do right now? Have we read the passage yet? Do we know what P addition is really? Not really. We haven't done that effort yet. So this is where now, this is when I actually read the passage. I will read the relevant section of the passage to make sure I can answer this question. It's what I think of as the treasure hunt. I look at my figure first and I say, okay, they're showing me an example of P addition there. I don't necessarily understand what's going on there, so maybe I should go back to the passage and read it. So I'm going to go back to the passage and read it. P addition, palindromic sequence addition, involves the creation of a DNA hairpin at the ends of gene segments, which causes unpairing nucleotides to form a single strand. Parent enzymes then add complementary nucleotides, generating a new palindromic sequence. Okay, so we're creating a new palindromic sequence when we do this, and we're using, so let's go back to the figure now and figure out what's going on in that thing. So it looks like in that top one we had CT, and then it took from the complementary strand the AG. So we kept some of those, the nucleotides that were on the top, and we pulled up two additional ones that were in reversal of the original other strand. Okay. So we go over to this thing and see, let's see. Suppose the last 6 nucleotides, blah, from a palindromic sequence on the 3' end. If the original double strand was cleaved 3 nucleotides from the 5' end. So, sorry, I think they're doing trash. Where are we going to stop, or where are we going to split this thing? Right down the middle. So we're going to split it here. So this part is gone because we're doing the P addition on the 3' end. So what should my palindromic, or what should my answer start with? Should it start with GAT or CTA? Remember, use the figure as a guide. The beginning of my sequence is what was already there or something new. Because this diagram says CT, and that was already there, and then to that I pulled the AG from below it, and that attaches to it. So am I going to use what's already there, or am I going to do something complementary to it? So is it going to be GAT or CTA? It's got to be the GAT, because that was the set of nucleotides that was already present on the chain, and that's where we attach our P addition piece to. So to use a very similar, well, first off, which answer is going to get rid of because of that? C and D can't be correct. So let me write my GAT that was on there ahead of time. So on my scratch paper, I'm just going to reproduce that figure. And here, what's my other strand going to look like? What's the complementary strand going to look like? C, T, A. And like in the figure, we're just going to pull that and pop it in its place. So what should my overall code be? Is it A or B? It's going to end up being B. And there we go. So it was a little bit of a challenge, but it is kind of a type 1 question. I would agree with that now looking at that. So, and we use the figure or the passage more to do this. Was the passage super helpful or was the figure more helpful? Really, it's the figure. If we just do the same thing the figure showed us, then we get the right answer. So is the passage super useful? Not so much in the sciences, not as much with the sciences. Sometimes it is, but most of the time, not so much. All right, how about the next question? What kind of a question is this one? One, two, three, or four? One way to figure it out is, does this ever show up in the passage? Did we see anything about this? Dissociation constants and stuff? So that means, what must it be? A 1, 2, 3, or a 4? Remember, figure 1 or type 1, that's me remembering something that I have to use to answer the question. Type 2 is when I'm giving you information, have to go to it. Is this coming out of my brain or out of the passage? This is a brain question. This is a type 1. I'm going to have to answer this from what I know about antibodies. So we got antibodies attacking bacterial strain, or against an early type and then a later type, weeks after the infection. And the dissociation constant is going to increase or decrease or stay the same. So again, we've kind of got this greater than, less than, equal to scenario. So what answer is probably not right? A is probably not right. Again, I'm not willing to outright eliminate it, but I'm definitely not going to consider it right off the bat. And here's another strategy with this one. See how it says slightly and much larger? Can you really make those kinds of distinctions? Probably not. I mean, that's pretty subtly different. It would have to be pretty egregious for me to know that there was going to be a slight versus much larger in something. So I'm also kind of discounting those. My gut is already telling you the answer is probably D. So let's verify that I think the answer is probably D. What's going to happen to the association constant weeks later? Do our antibodies get stronger or weaker over time? They get stronger. They're better at their job, which means their association constant is going to go up or down. The association constant is going to go up, which means my dissociation constant's got to go down because they are inverses of one another. If you didn't remember, Ka is the inverse of Kd. So I think we're vindicated in saying the answer is actually going to be D, because if the association goes up, then the Kd is going to go down. Now, could you have used the passage to answer this question? The answer is yes. Did anybody catch it when they were going through? It's okay if you didn't. And in fact, I'm happy if you didn't, because that means you didn't read it that well. But look at the fourth paragraph. Affinity maturation, that's right. So the hypermutation increases even more, and that happens with activated B cells that develop a greater affinity for their antigen through somatic hypermutation and are selected based on their affinity for the antigen. So it's in there, but we didn't have to see that because that's honestly something you should know anyway, is that, you know, this is more of a memory question because that's something you should know about this. And like, if you did know it, I would fully expect that you would probably want to go figure that out. That's why I say this is kind of a 2 question, because it's kind of a 2 question in the sense that if you don't know this fact, then you got to go back to the passage to figure it out. But if you know the fact, and I definitely want you guys all to know that fact about the immune system, then you should be able to just answer it this way as well. Alrighty, how about our next question? What kind of a question is it? I would actually put this as a 1, because where did I see each of those things? The passage, with one exception. Which one didn't I see in the passage? Not like the phrase. I didn't highlight anything that said gene fragment rearrangement. So I don't know about that. But it's in the passage and they define them all in there. So that's how we should approach it. I would call this a 1 since it's in the passage and it's like just straight definitions in that passage. We noticed that. So which Roman numeral do you want me to attack first? Good, you remembered. Go after Roman numeral 4 first because that's the one that shows up twice. So affinity maturation. I'm going to go back and I'm going to read about affinity maturation. In a process known as affinity maturation, activated B cells that develop a greater affinity for antigens are selected for and based on their affinity for antigen presented on the surface. They have a weaker affinity for the antigens as those that have a weaker affinity for antigens are signaled to apoptose. So is affinity maturation increasing the variability of my who's the moetsits of my B cells, of my antibodies? No, it's not. According to this, it's a process that's based on selection. So if anything, it's increasing or decreasing the variability. In a way, it's actually decreasing the variability because I'm removing certain types of B cells. The more so we're honing it in now. Our B cells initially start out all these different versions, and then we're weeding out, we're apoptosing those that don't work. So in a way, we're actually decreasing our whatchamacallit, variability. So what do we think of 4? Is it good or should we get rid of it? Yep, let's lose 4. Which answers go away? C and D. All right, so now what Roman numeral do I have to look at? Roman numeral 1, because I know 2 and 3 are both true. I don't even have to go read about those things. I have now proven a fact that I have a proven fact that I could use to answer other questions if I wanted to. So we should go back and read about somatic hypermutation. So somatic hypermutation says another mechanism for Fab variability is okay, I'm already done answering this one. Is 1 true or false? Like straight up says it. It's a mechanism for increasing your antibody variability. So, okay, it's true. So what's our answer? It's got to be B. And notice every step of the way, what have we been doing with our passage? We've been learning more about it or learning less about it. and we're doing it in just like this little piecemeal way. Is it as intimidating to do that? Not really. I mean, when you're doing it in these little chunks, it's more of a bite-sized thing, and it doesn't feel as like overwhelming when you're going through that. Okay, so now we got one more question to answer. Which B cell processes would one expect to occur in a germinal center of a lymph node? So again, we've got a Roman numeral question. What Roman numeral would I want to attack first? Two or four, it doesn't really matter. So let's see, I'm gonna go after two. B cell maturation. This is actually your knowledge that has to answer this, because does it tell us where this stuff happens in the passage? I mean, we're talking about B cell maturation, but does it say where this happens or just describe it? And if you're reading the second paragraph right now, that's a smart thing to do. So is it telling us where or just telling us what? Yeah, it's just describing B cell maturation. So we're gonna have to dig out of our memories where that happens. Or I could just skip 2 for now and move on to 4. So why don't we do that? Like if I was on a test, I might go like, I don't remember that. Let me look at 4 then. Productive rearrangement of VH, DH, and JH gene fragments. So where does that happen? Does it happen in the germinal center of a lymph node? So what is productive rearrangement? Here it is. It's that same paragraph. Well, crap. It just describes it again. Okay, now I'm forced into asking myself, where does this happen? Does anybody remember where B cells mature? Does anybody remember why they're called B cells in the first place? The bone marrow. The bone marrow. And why are T cells called T cells? Where do they mature? Not the thyroid. It is thigh something, not thyroid, thymus. There we go. The thymus. So that's where these things mature respectively. So what do we think of 2 or 4 actually? Are they good answer choices? Is a lymph node part of the bone marrow? So keep them or get rid of them. Get rid of them. And so that means answers B and D go away. What's my last Roman numeral I have to look at? Roman numeral 3. So that one's somatic hypermutation. So what does the paragraph tell us? Is that going to be something that would happen in a lymph node? Somatic hypermutation occurs after a B cell encounters its specific antigen and involves a greatly increased chance of mutation in all CDRs. What happens in lymph nodes? In fact, why does the doctor look for your lymph nodes when you think you're sick? Which, wow, I just felt one of mine and it's swollen. They're getting swollen because what's going on in there? It's getting flooded with white blood cells because it's encountered a antigen. And what are white blood cells? Oh, B cells are white blood cells. So does it seem likely that 3 is true then? Three, keep it or get rid of it? Yeah, I'm gonna keep it because that seems pretty true. So my best answer is answer choice A. So let's think back in the process that we used then. How much did we actually use the passage? We use it a lot or just a little bit? Really just a bit. Not really a whole lot of like hardcore. Like there's only 2 places where I actually read anything of any note. The first 2 paragraphs or the last 2 paragraphs, which ones were more useful? The last 2. And the third paragraph, did we ever read the whole thing? Did we need to read the part about N addition or junctional flexibility or any of that? No? Like, in fact, all in all, when all is said and done, what I would say that you needed to do was reading about P addition and reading this whole paragraph. Unnecessary to read any other parts of this, unless, of course, you're going after the, well, no, actually, that would be it. So now, what's my point? Why am I stressing this so heavily? One of the things that I see happen to students so very much is getting overwhelmed by the passage. If it's a very dense passage and you're trying to make sense of stuff and it's not necessary to understand it, it's a waste of time. So I want you to learn to skip over things. You don't have to focus on everything when you don't really need it. It's actually somewhat the same as cars. In a lot of cars passages, there's certain paragraphs you don't actually need to know. And so there's no reason to really dig through it hardcore. It's just, note what's there and move on. Don't get overwhelmed with the density of the passage if it's not going to end up doing something for you at the end. So, and this is very typical of science passages to have this nature of not really needing most of the passage, especially the more figures a passage has, the more likely it is you're going to use less of the passage itself. It's not perfect, but it's usually the case. And then you want to have some variability in your process too. When you're doing science passages and they have no figures, what's a good way to tackle it? What strategy might you try? A no figure science passage. Well, that number one, yeah, I would totally skip it for later. But when I actually have to do it, am I going to treat it like a science passage? Yeah, I would totally use my CARS method on it. And I do. With no figure science passages, I go at it like it's a CARS passage instead of a science passage. And you'll see that a lot actually in Psych Soc. There's a lot of passages in Psych Soc that have no figures. And honestly, I just treat those just like I do with CARS, and I do them the same way. For figure heavy ones though, I cherry pick. And if you want to give this technique a name, let's call it the cherry picking technique, because all I'm doing is I'm sitting there and I'm pulling this little bit and this little bit and this little bit, because I don't really want to read this whole darn thing. It's not going to help me. It's not going to answer questions. And at the end of the day, what do you got to do is answer questions.